In the last lecture, we got a first glimpse of the unique and unusual properties of water, its polarity, based on the high electronegativity of oxygen, and its ability to form hydrogen bonds with up to 4 other water molecules, 2 as donor and 2 as acceptor. You'll remember that all 4 of these bonds are only realized in ice, while liquid water is more densely and more randomly packed. The molecules have too much thermal energy to stay in one place, and the molecules jiggle about and form hydrogen bonds only fleetingly whenever 3 atoms involved are aligned correctly. Today we want to have a broader look at how water organizes molecules and the supramolecular structures in a way that makes life possible. We will look at the solubility of molecules in water, as well as the influence of water on non-covalent interactions. We will then look at the ionization of water and the basics of acids, bases, and buffering. It is difficult to overstate just how central water is to the chemistry of life. Chemistry happens in all kinds of solvents, from benzene to liquid ammonia and everything in between. But in biochemistry, water determines everything: what shape molecules assume, whether they are soluble, how they interact with each other, and how they react with each other. The characteristic shape of all macromolecules and supramolecular structures of life, DNA, polysaccharides, lipid membranes, and proteins, is largely determined by the response to water. So, what is it that determines how molecules behave in an aqueous environment? The polarity of the oxygen-hydrogen bond, as we have seen, makes water a dipole, and is the reason why water forms extensive hydrogen bonds with itself. However, we have seen that similarly polarized bonds exist in biomolecules in the functional groups. For example, this molecule, the amino acid alanine, contains a carboxyl group and an amino group, both with strongly polarized bonds. Such polar groups give the whole molecule polarity, and the stronger the overall polarity, the more soluble a molecule is in water. Hydrogen bonding between water and polar groups is the reason for solubility. Polar water molecules align themselves around polar molecules, and the formation of hydrogen bonds makes the solvation of such molecules, that's what the process is called, energetically favorable. If the solvent is water, we also speak of hydration. The molecule is solvated or hydrated if it is surrounded by solvent or specifically water molecules. Something similar happens with ionic compounds. Take a simple inorganic example, in this case a sodium chloride crystal. The sodium cations and chloride anions are held together by electrostatic forces, and these are quite strong. However, water molecules are able to disrupt this lattice by solvating the ions and surrounding them with so-called hydration shells. There are typically several of those shells in layers around the ion or other solvated molecules. Note how the orientation of the water molecules reflects the polarity of the ion, although there is quite some freedom. So even though in the crystal the electrostatic attraction between the ions was quite strong, the formation of this hydration shell and the fact that the attraction between positive and negative charge is much weaker in water than almost any other medium together means that solvation is favored over the crystal lattice, and the ions dissolve. We should bear in mind that this kind of organization of a hydration shell is energetically favorable, while the hydration shell of a very apolar molecule, like benzene here, organizes the water molecules around it in a very specific arrangement. As the figures on the right, which come from computer simulation, show, the orientation of water molecules in the first hydration shell in yellow is much more organized than the bulk water around it. As we discussed in the last lecture, this organization means a loss of disorder or entropy, and is thermodynamically very unfavorable. Another well-studied example, if not really from the world of biochemistry, is that of methane hydrate. Methane is completely apolar, and if brought together with water under pressure, it forces it into this beautiful, very regular cage, where it seems that the water tries to maximize the formation of hydrogen bonds around a molecule that cannot form them itself. So let's summarize. Highly polar molecules with a large proportion of ionized or polar groups are water-soluble or hydrophilic because they're able to form multiple hydrogen bonds with water molecules. It is possible to estimate how water-soluble a compound is going to be on the basis of how many ionic, polar and non-polar groups it has. Non-polar molecules are hydrophobic or water-hating because they lack polar groups that could form hydrogen bonds in water. They tend to minimize contact with water by aggregating. This reduces the entropic effect of water molecules being more highly structured and less mobile in the water layers surrounding non-polar molecules. Now, some of the most interesting molecules in biochemistry are not clearly in either camp. This is true for pretty much all proteins. Much of their function is determined by the specific distribution of hydrophobic and hydrophilic, as well as polar and charged regions. We call molecules that have both polar and non-polar regions amphipathic, or sometimes amphiphilic, meaning that they both hate, or fear, or avoid, and love water. In the narrow sense, we call molecules amphipathic if they have distinct groups with opposite properties, such as this molecule, a membrane lipid. It has a polar side, which in this case we also call head, with multiple ionic and polar groups. It also has two long hydrocarbon chains that give it a long hydrophobic tail. What's interesting is now what happens when this molecule comes in contact with water. Let's look at a simpler molecule, one of the two fatty acids that are part of the lipid we've seen before. As we have discussed before, the hydrophobic tail of this molecule imposes a particular order on the water around it. Water is unable to interact with these non-polar hydrocarbon chains. The water molecules that directly contact the hydrophobic tail try to make the best out of the situation and form what we think is this cage-like structure around the hydrocarbon chain, so they at least keep hydrogen bonds with each other. They have a lot less freedom to move around and hydrogen bond with other water molecules, and this constraint is the basis of the hydrophobic effect, a decrease in entropy or disorder that is unfavorable. The same is not true for the hydrophilic or water-loving head group. Hydrogen bonds can be formed here. To improve this unfavorable situation, these hydrophobic tails associate with each other. In this scenario, proportionally more water molecules are free to hydrogen bond without constraint, which increases the entropy of the system and is therefore favorable. The tails are shielded from interaction with water. On a larger scale, amphipathic molecules aggregate into micelles, and in a lab environment sometimes into monolayers at the air-water interface, as well as into vesicles. Note how in vesicles we have a double layer or bilayer of these molecules, with the polar heads at the surface and the hydrophobic tails tucked away and facing into the bilayer sheet. These are obviously schematic models. The reality is probably a whole lot messier than this. Tune in for part 3b on the dissociation of water, acids and bases.